good evening. Welcome to July Cafe Scientifique. Tonight we're here for New Zealand's bird malaria and why it is not always a bad thing. The largely unseen drama of malarial parasite infection, parasite burden, and sometimes death is playing out all around us in New Zealand within our native, endemic, and introduced bird species. Our speaker for tonight is Dr. Rosemary Barraclough, who is one of the Auckland Museum Institute's counsellors for her. She must have many sons. Rosemary is an honorary academic within the School of Environment at the University of Auckland and is a research associate with the Auckland Museum and the Field Museum Chicago. So without further ado, can you please put your hands together and welcome Rosemary.
Thank you, Scott. So, I don't know, has anyone here had malaria themselves? No. Well, I'm pleased for you. Just to let you know, malaria is a really old organism, and most people think of it in terms of humans. And this is an example, just for your interest. I've dropped a round of images out, and I'll circulate, and if you guys want to ask any questions about them later, I'm here. But these are human red blood cells. These pink, blobby, characterless things. Here is the killer falciparum, and that's the one that can become deadly before it's even visible in the peripheral blood. And this is what you'd likely get if you went to the Solomons or Papua New Guinea, and we're really unlucky. That's Vivax, and that reoccurs. But in comparison, these are bird red blood cells. You can see they look like green grapes with enormous seeds. And that's why if you want to do DNA work on any birds, you just need a few drops of blood, because every single red blood cell is just loaded with DNA, as opposed to our pathetic little corpuscles that carry around hemoglobin. So our red blood cells lose their nucleus as they develop, whereas bird and reptile red blood cells do not. Malaria is very, very old, and it's really interesting because it has 3 types of DNA. And by the way, it's not a bacteria, and it's not a virus. It's an apicomplexan organism. It's a protozoan, which means it's single cell, but it's got a defined nucleus. And it's also got DNA in its nucleus, DNA in its mitochondria, like we have mitochondria in each of our cells. And then it has this other organelle that seems to have been derived from a plant-like organelle. So it's got these 3 types of DNA. And this third organelle is involved with something to do with the metabolism, one of the metabolism pathways. So it's an unusual organism. And so we think of it primarily affecting humans, but it also affects rodents. I'll take these off so I can see you guys. It affects rodents, it affects apes, a variety of primates. It affects snakes, lizards, and birds. And the vector for each of these malaria is a mosquito, except for the vector for some of the lizards, where it's what's called a sadfly. It's not what we call sadflies in New Zealand, and some other biting organisms. So the life cycle of malaria is hidden from us. It spends its entire being inside one of its hosts, either a vertebrate like you or me, or a bird, or inside its insect host, its blood-feeding host. And human malaria and all the mammalian malarias are only vectored by Anopheles mosquitoes, and we do not have them here in New Zealand, which is absolutely fantastic. With global warming and international trade and second-hand tyres reaching the country, who knows, maybe we welcome that as a new species. In the interim, we should be really pleased. But not every Anopheles mosquito can vector every species of malaria. So I do not understand how early scientists worked out the life cycle of malaria. The name malaria comes from ancient Rome, where they associated illness with the pestilent fumes of swamps. And so they thought by breathing in these, foul smells, you were getting ill. It wasn't until 1880 that Laveran, a doctor with the French armed forces, identified it as a parasite. It must have been representing decades of hard work, because this parasite, all its stages look so different from every other stage. And within a vertebrate vector such as myself, it has more than one type of cycle, and it looks different in each of these cycles. Then the infective stages, once they're sucked up by a mosquito, they transform into gametes and undergo sexual reproduction inside the mosquito. So this organism cannot reproduce, get into another vertebrate host, without doing the wild thing inside a mosquito. So this is a truly complex organism. It doesn't stop there though, because once they're transformed into gametes, the female protogamete and the male protogamete has been sucked up at the same time. They're transformed into gametes. The male protogamete divides up into these flagellating organisms that swim sort of like human sperm. Hopefully they find a female and gamete, and then they go through several developmental stages before an infective stage, which looks totally different, reaches the salivary glands of the mosquito. So that mosquito has to, each mosquito has immune defences, just like we do. So a whole bunch of mosquitoes around the world may accidentally suck up a whole bunch of malaria female and male protogametes. But once they've gone through that sexual reproduction inside the gut, they can't actually survive through the next stages and migrate to the salivary glands. So a mosquito and the malaria has to have evolved long enough together for it to be a competent host. And I was studying malaria in Madagascar in birds, and I was being bitten by a variety of organisms, and these birds were highly parasitised, not just by malaria, but by microfilarial worms that were hideous things to look at, interesting, and trypanosomes. And so I thought I better check my own blood. And so I took my own blood and made a blood smear and looked at it under the microscope. Absolutely clear. Our immune systems are fantastic things, but it also means that, a bird's immune system means that it may be resistant to one malaria, but not the other. And the mosquito that happens to feed on that bird is resistant to one malaria, but not another. So there's all these complexities and long evolutionary time. So we may assume that it's always bad, and yet if we go out and study birds, and for instance, netting stations in Vermont, which may sample over 1,500 birds in a season, and they can detect at peak times enormous prevalences of malaria and closely related organisms with a similar life cycle. And these birds are not sampled because they're sick and struggling or unwell or came to a vet. They were caught out in the wild and not detectably different from any other bird that was caught without it, and was released. That we simply detected it because we went out and captured it. And some of those prevalences can be as high as 80%, or they can vary. Some species seem to not be very they seem to be able to resist malaria really effectively. But on the other hand side of that spectrum, you have places like Hawaii, where you've had these honey creepers. The top one's an Evie, and that's the one that's evolved with its beak shape just to nicely fit the shape of the flowering lobelia trees. And then that's a papane, and that's a mokane. So of these three honey eaters, this top one, in captive trials, more than 90% of them would simply die of malaria. If one survived, then it could be resistant to the fungal infection. The upper party, more than 60% in these captive trials died, and more than 60% of the amakihi. So there's a whole, Hawaii, like New Zealand, has a rat problem. It has all the same sort of, instead of a possum or mustelids, they have the mongoose. So they've got all the same problems as us, but they also have this vulnerability to introduced avian malaria. There were no competent mosquito vectors in Hawaii, and either the malaria was present in migratory species, maybe along the shore, but it wasn't able to jump into these passerines, or an introduced species brought the malaria. The mosquito, Quinta fasciatus, it's a culex, was brought to the country, and it was thought it arrived in water barrels on sailing ships. And that provided, the links to be, filled. So the amakihi, the little green one, and I'll pass these pictures around, although the ones still capture from higher altitude, and we're talking about a 1,200 approximate boundary before you have, it's sort of fairly safe for these really vulnerable species. The ones caught high may still be fairly vulnerable, but there is a resistant lowland population of the amakihi that's been developing over the last 10 years or so. So that's really heartening news. However, with global warming and a lot of these remnant populations stuck at high altitude, they're going to become increasingly vulnerable. And when I was last in Hawaii a few years ago, Carter Atkinson told me how in Kauai, one of the older islands, and they have this Kokei, I think it's called, natural area at the top, and they've been suffering drought, which meant the flowing streams were pooling, and it was just allowing these mosquitoes to get slightly higher and higher perches, and they were getting to the stage of considering going out and just trying to trap as many birds as they can to put them into a captive area where they can be protected, netted against the vectors of malaria. It's extraordinarily tenuous. So that's what it can be like if you have a totally naive population. But in New Zealand, Hawaii has got this one variety of malaria. There's something like 55 avian malarias in morphotypes around the world, much more diversity than that if you're talking about genetic lineages. And that's just in Plasmodium, the true malarial parasite. The first cousins of malaria, there's also another great swathe, and the second cousins of malaria, all organisms are commonly encountered in Madagascar, are also prolific. In New Zealand, we had some early research done by a fellow called Dore in the 1920s and Marshall Laird in the 1950s. Does anyone know Marshall Laird? He was around for a long time. He was a great talker, so if you met him, you would have remembered him. And he had a distinguished career with the World Health Organization, researching things like river blindness in West Africa. Anyway, so they surveyed for malaria, avian malaria in New Zealand, and I'm using the term avian malaria really loosely because, of course, that is the disease, representation of it, and it's the parasites having an effect. But we're talking about the parasites that cause the malaria, Plasmodium. And their searching was mainly focused on introduced species, because even back then, native species were a little bit inaccessible. They were restricted to hillier areas, or there was permitting issues, etc. Later, following that, however, in the 70s, a survey of penguins discovered the second cousin of malaria. It was called Leucocytozoan penguins. So in the world, the species of Leucocytozoan is named after the Fiordland rockhopper penguin in New Zealand. But as far as malaria itself, Plasmodium goes, we've got approximately 8 species. And this is a little bit small to see. But basically, this lower area is introduced hosts. This work was done by New Zealand scientist John Ewan, who's based over in the UK and Italy. And the top, some native species. And you can see that the prevalences tend to be greater in the introduced species. And for instance, up to just over 80% of some introduced species such as blackbirds can be found to be carrying it. And in this graph, he has up to 40% of blackbirds. Virtually every blackbird I've looked at through microscopy has been carrying some sort of malaria. But New Zealand endemic species don't seem to carry an awful lot of it in easily detectable levels. The greatest prevalence, apart from one example, is in saddleback. But although malaria in New Zealand has been associated with the death of several birds, including South Island saddleback, and captive dotterel in association with avian pox, in the death of mohua in Orana Wildlife Park, about 5 out of 8 captive dotterel, mohua. There's also great spotted kiwi, brown kiwi, and hihi, and occasionally introduced species who have been found dead, and they are found through necropsy to be carrying malaria. Sometimes, is it the chicken or the egg? Was there something else that they lost control of the malaria, or was the malaria out of control and killed the animal? But despite those deaths, we also have prevalences such as the North Island saddleback, and in these surveys we found between 2 and 10% of birds carrying one of 3 different lineages, representing 3 entirely different types of malaria. None of these birds were sampled because they were ill. We were simply surveying to try and get a handle on what role malaria was having in the system. And a bunch of other birds. 17% of these tui that a student of mine and I sampled were carrying malaria, and so forth. So yes, there has been death, and yes, a couple of those events have been quite dramatic, but it is not like the situation with Hawaii. And perhaps one reason is that we potentially have at least one native malaria. So in the bellbird, over the 2007-2008 season, we did some sampling. Whoops, I should have probably got that one, that's got the pictures. And we discovered approximately 50% of the male bellbirds on Terry Island had visible malaria in their peripheral blood. And 3 years later, 41% was detected. So it was nice to have that repeat survey at a similar level. During the 2007-2008 year, we also found 25% of the male bellbirds at Tawharanui nearby also had visible malaria in the peripheral blood. And these birds, it was a pretty cool result because these birds were not obviously sicker or iller than any other bird. And again, we only sampled them because we were out there meeting to see if there was malaria on the island. At the time of the first survey, I also looked at thin blood smears from 168 other birds across different species and did not find any at that stage, not visible in the peripheral blood, though it could have been there detectable by PCR, which is much more sensitive. What's also really cool is about this malaria is that when we looked at the sequence, it was really similar yet distinct from 2 lineages that were found in other honey eaters, one from Queensland, the other from the Papua Australian area. And the 3 together were really distinct from others around them in the tree, which indicates that this is evolved from this part of the world, and these are lineages which have been detected mainly, at least thus far, in the honey eaters. Now, this lineage Bell01 in New Zealand has been picked up in some other species, and some of the sampling done for that was done on Tiri, where there's so much of this that it's not too surprising that it has been picked up in some tui and also in a few saddleback. Again, not because those birds were ill, but because we were out there surveying and colleagues were out there surveying. Now, just because, however, that you go out and you look at a bird, you find it's got some malaria, but it seems perfectly fine, it doesn't mean that there isn't a cost to the bird. It's a burden to be carrying this extra parasite. And it does mean that if that individual becomes starved or stressed or otherwise compromised, then potentially they'll lose control of this malaria. This may have happened with this event at the Auckland Zoo with this young bird who was 2 years old. It had been shifted to another captive area, and suddenly it lost a third of its body weight and it started presenting some neurological symptoms, and it was absolutely riddled with avian malaria in its peripheral blood. I mean, at least half of every red blood cell had a gametocyte or some early stage of malaria in it. It was so riddled, there were very few mature stages to actually see. It was just full of malaria. So that bird was euthanized. Whether it was definitely the same type of malaria as the Tiri, and that Tiri malaria is found throughout New Zealand, but generally at lower prevalences than we have found on Tiri and at that time on Tawhanui. Another interesting thing, I won't try and find a graph for it, is that a student of mine that went out to see whether the capture method you used affected your results when you were surveying for malaria. So has anyone here been to Tiri? Yeah. Yep, everyone. You know the feeders. And so you know that when birds enter those feeders that were meant for the hihi, but the bellbirds are stroppier and smaller, and you can't keep a bellbird out from a hihi feeder. You can actually trap birds in there very easily just by closing the door, putting a piece of string there, closing the entrance. So David and Marta, some student, tracked birds in feeders. They focused on male bellbirds just to try and simplify comparisons. And they also netted birds. And they netted birds a little bit away from the feeders. They banded every bird, and quite a few were already banded from a previous study. And so there were no recaptures. And so there was no confusion about capturing the same bird in both methods. And they found that the birds in the feeders were fatter, a lot fatter than the ones in the nets. All the prevalence across the whole group there was no difference. And so no evidence of any, and when I say prevalence, that's the proportion carrying visible malaria. But all the intense infections, all the ones that had a big parasite load were caught in the nets. So does this mean that the fat birds were so nourished and full that they were controlling the malaria better? Or does it mean that the birds that were riddled with it, well, they weren't riddled, but they just were carrying bigger loads? Not all the netted birds were carrying huge loads, but all the ones with big loads were caught in nets. Maybe that could potentially have stopped those birds penetrating that dominance hierarchy that works around the feeders, where they're protected by these, birds that have got a purchase. So it's a bit of a chicken and the egg, but it did show that according to how you catch your birds, you're going to get a different result. If not with prevalence, definitely with the degree of infection. Another interesting question in New Zealand surrounds vectors. So, assuming, and there's potentially the chance of there being another, at least native one, malaria, and that we found a single detection of a Plasmodium in Kokako that was just opportunistically sampled during translocation, and that bird was fine, but that lineage hasn't turned up again. So maybe the story isn't complete yet. But the vector story is a little bit of an unknown in a big way, because the mosquito that arrived in Hawaii, which is the smaller one here in these pictures, is in New Zealand. And people have speculated about it increasing the transmission of malaria in our birds. We don't know whether this mosquito is a competent vector for this Barraclough 1 lineage or all of the other species we have here. We don't know. Assuming Barraclough 1's native, then maybe our most common mosquito, which is Culex purvigilans, is the vector. We don't know. So there's some nice unanswered questions here. It is a bit of a worry that a student of mine did a survey at Auckland Zoo. They were interested in updating knowledge about what potential vectors were on site. And the results differed from a survey done 13 years previously. 13 years previously, there was introduced Aedes notuscriptus. That is not an avian malaria vector. It was tested over in Australia years ago in an historical study. But it could potentially vector all sorts of other nasty things like dengue, etc. So that's something to look forward to. But notuscriptus has been here for years. It's the one that bites you with white little stripes on its legs. So, yeah, and that can breed in the tiniest little pockets. So if you want to avoid having mosquitoes around your house, empty any standing water. At the zoo, they give the animals lots of nice, clean, fresh green foliage to eat, and they use these big water containers to keep them fresh, like huge vases. The pride lands, great big water storage things, had no mosquito larvae. Near the primates and other places, they were loaded with mosquito larvae, and the difference was pride lands emptied them once a week. So just keep your place free of little pockets, standing water, and you won't have this mosquito issue. But that notuscriptus can breed in the axils of bromeliads. Yeah. So water your bromeliads. If you flush them out, you can flush them out. But yeah, think about what you plant. But the other one that had been previously detected there was the Culex conquifasciatus, which is that competent vector in Hawaii. When we went back and resampled, we didn't find any native mosquito larvae there except for a little one, just like one or two larvae in the axil of a bromeliad or some sort of introduced species near near some of the primates, and that was the only sign of the native mosquito we set down. Everything else was the introduced conquifasciatus and the introduced Aedes notuscriptus. So those, when a bird is in a cage, it is like a sitting target, isn't it? And so it is a risk having areas where you actually have active breeding, sites in proximity to these captive birds. So that was an interesting result, and I hope it was useful for the zoo. So that's another question that's asked about this. And the other interesting thing I think about our malaria in New Zealand is that malaria can be a model for human malaria. Some of the earliest research done on human malaria was using a bird model because it was much ethically nicer to use something that appeared non-lethal to birds as opposed to the lethal one on humans. And so we can use that for things like looking at the sex ratio of those pre-gamete stages and look at the evolution of virulence using the characteristics of our fauna. So I think it's a really interesting field, and of course when you're talking about a parasite that's potentially going to be affecting the energy levels and etc. of its host, then you're going to be looking at the fact that this is going to be affecting all of its life stages potentially, and so really it's an aspect of research that can be added to many other fields.
Alrighty, can we bring our hands together and thank Rosemary for... If you've liked this video, please make sure you like and subscribe so you can check out more of our cafes in the future.